Okay, so this is the last PowerPoint of the semester and the last material, and test 5 will be next week on the syllabus. If we were face to face, we would be doing presentations next week, but that isn't going to happen. At least this semester, I just don't haven't figured out how to get this all together, and this is um, how to get it going here, maybe in further semesters. But so I graded the tests. Hopefully you've seen them. Make sure you look at how I've set up the blackboard. Uh, there will be more columns than grade and then actual grades. The actual grades will come from the columns that say test 1, test 2, test 3 core, and test 4 core. So test 3 core and test 4 core include your homework, and so each one of those each test is worth 100 points. The next test is going to be similar. There'll be a worksheet and a test, and they'll be combined for a test 5 core. So the total score for the semester will be those 5 scores, each one 100. So it's 500, and then divide by 5, and that'll tell you your percentage score. Okay, hopefully you'll figure it out. Make sure what's on the PowerPoint, on the Blackboard, is what you think it should be. Okay, if you have any questions, please get a hold of me. Okay, so the idea is that I will probably put out the test for this to Monday, I think. And you probably have until Wednesday to get it done. You guys have been doing real good with it. It looks like all of you been doing what you should be doing. Don't like saying this, but you always a little suspect of people going on. I can't be there to supervise you or proctor you. So, but it looks like from your answers, it looks like there are answers that you are actually put your notes away and have take the test, and I really appreciate that. Okay, that's really good. Now, all I can tell you is that everybody looks good. Okay, so we'll get through this semester, and you guys be able to move on. That's cool, and hopefully you'll get into your vet program or nursing program and move on. Very cool. All right, so let's get going. So this would be chapter 17 in your book. If you want reference, actually, it's not a bad reference that to take a look at. If you're kind of confused about something, you can go there. There's all kinds of resources these days. You go to Khan Academy and etc. and try to get other things. One thing is when you on a test, if you use something that's from an other resource. I've had, on the last test, I had people use things that I never talked about. Okay. If you do that, reference it. Okay, so tell me where you got it from. That way, I'm a little bit more confident about giving you the appropriate response for your answer. Okay, like I had several students. Well, I don't know several, but a couple of students said functional proteins were storing nutrients, and I never said that. And where'd you get that from? Okay, so it's better to tell me where you got it from than me just go, where'd you get that? Okay, because to me, when you say that, if you say something that I haven't seen before, it seems to me that you're just kind of fishing in the internet for something to get the answer. So be upright, upfront about it, so that so we can so I know what you know. And I mean, that's what's about. That's what's about this class. Is I want you to walk away with some things that are I think are important. Okay, nucleic acids and protein synthesis. Okay, so the final macromolecule is nucleic acids. And we're going to learn about their structure, just like on carbohydrates, lipids, and proteins. And from their structures, we are going to try to and look at their functions. And there's a couple main functions that nucleic acids have. They have all kinds, but a couple that we're going to look at are replicating themselves, making more nucleic acids. And nucleic acids is the information to decide how proteins are made. So proteins are the most important molecule in the cell because they have so many jobs. How do they get made? Where's the information for them to get made? And it turns okay. So nucleic acids involved with hereditary. So so this is genetic stuff. Okay. So they are the mechanisms for genetic information. So it's our DNA. So if you have Ancestry.com and you get the DNA, if you get 23andMe or whatever, what you do is you spit into a cup that has your DNA in it. Okay, and that DNA tells your body how to make tells the body the proteins that it makes in your body. Okay, and so and other things. Okay, so and it gets past your DNA, gets passed through the generations. Okay, and we'll talk about how that's done a little bit. Okay. There are two kinds of nucleic acids we're going to talk about. One is DNA, which is deoxyribonucleic acid. Okay, so D stands for deoxy, ribo, N stands for nucleic acid. So ribo will be related to a sugar that we talked about already, and when we talk about carbohydrates, deoxyribose. Nucleic acid means it's found in the nucleus, and it's acid, and we'll learn about that because it has a phosphoric acid on it. Okay, it contains the genetic information to make DNA. To make proteins. All right, the other guy that we talk about will be RNA, which will be slightly different. One of the differences is that instead of having a deoxy, it has a ribo. That's why it's R. And again, it's found in the nucleus, and it's acid. There's several different kinds of RNAs. There'll be messenger RNA. There'll be, which is called mRNA. There'll be transfer RNA, tRNA. There's another type that we won't talk about called ribosomal RNA, rRNA. But they're all involved with the process of making, getting the information from DNA to make proteins. Okay, so let's look at the most basic subunit of a nucleic acid, and that's a nucleotide. Okay, so nucleotides are like amino acids. So nucleotides and nucleic acids are like amino acids, the proteins. So these are the building blocks of nucleic acids. So nucleic acids are going to be chains of nucleotides linked together. Okay, like a protein is a long chain of amino acids. A nucleic acid is a long chain of nucleotides. So we're going to look at the structure of nucleotides, just like we did look at the individual structures of amino acids. And nucleotides are made out of three parts. They're made out of a sugar, a pentose, a 5-ring sugar, a 5-carbon sugar, a nitrogenous base, which we'll talk about, and phosphoric acid. So we're going to talk about these three things on this guy, and then we're going to link these guys together to form this, the nucleic acids. All right, so these are the three parts of a nucleotide. You have your base, nitrogenous base, and we'll talk about why it's called base in a minute, because it has to do with this N right here. We have our sugar, and then we'll have our phosphate. The sugar is going to be really important in terms of this is a pentose deoxyribose or ribose, and it has 5 carbons. Okay, 1, 2, 3, 4, 5. And what we're going to do is, what you're going to have to know is, first of all, you're going to have to number the carbons, 1, 2, 3, 4, 5. Okay. The other thing, you're going to have to tell what each carbon does. Okay. So right off the bat, number 1 carbon is connected to the base. And number 5 carbon is attached to the phosphate. Okay, so make sure you write that down. So that's either for ribose or deoxyribose. Okay, so again, the parts, you have the pentoses, which are going to be deoxyribose or ribose. We'll look into their differences in a second. You have the phosphate, which came originally from phosphoric acid. So the A in DNA, and this A right here, is acid. So deoxyribonucleic acid. And where it comes from is the phosphate. Okay. And the base, this is a thing that's going to change. In amino acids, the side chain changed, and there were 20 different amino acids. For DNA and RNA, what's constant between in the RNA or DNA is the sugar and phosphate, but what is different between them is the bases. For DNA, there are four different bases. And for RNA, there's also 4 different bases. And you notice for DNA, there's CTAG, and RNA, there's CUA and G. So for RNA, it doesn't use T, it'll use U, and we'll see that how to use that in the future here. Okay, so the sugars. Okay, they're pentose, just like we studied in carbohydrates. You have your deoxyriboses and their Ds. If you remember about that, you don't have to know how that applies here. But that's what they are. And so let's look at them. So, so you have ribose and you have deoxyribose. Now, they're exactly the same, except the difference is at the second carbon. The second carbon for ribose has an oxygen, and for deoxyribose, there is no oxygen. That's why it's called deoxy. Deoxy means without oxygen. So it's missing oxygen. So one of the things you're going to have to know about these sugars is, again, what is the role of each carbon. So the number 1 carbon, we don't have it here, but it's attached to the base. Number 2 tells if it's RNA or DNA. If it has an O, then it's RNA. If it doesn't, it's DNA. And then number 5 we talked about had a phosphate on it, not in this diagram, but in the previous diagram. So right now we have the roles for 3 of the carbons. First one binds base. Second one tells the difference between RNA and DNA. Number 5 binds to phosphate. Okay, so a little bit about the phosphate. The phosphate is bonded to carbon number 5. And actually, I mean, it turns out, a lot of times we just kind of, when we talk about DNA, we just kind of just talk about it, all right? But this thing here, phosphate, is really, really important in terms of growth, okay? And I'd love to talk more about it. But I mean, what I mean by growth is how species, and I mean not species, but how things can grow, plants and animals can grow fast and slow. It has to do with phosphates. But we won't talk about that right here. So yeah, it's combined to number 5. If you notice it has a negative charge on it, that means it can be attracted to water real well. It can be polar-like. Okay, so we're gonna learn. See in a minute. Is that turns out this guy is kind of like soap, has a polar head and non-polar tail. Okay, and that's gonna kind of help how DNA forms and RNA forms. Okay, and similar to everything we talk about. Actually, when we talked about phospholipids. Okay, a phospholipid had a polar head and non-polar tail. And if you remember, the polar head interacted with water, and a non-polar tail had two kinds of interacted with another non-polar tail over here. Okay, it's made a little laser thing going all over the place. And there were two interactions. There was side-to-side tail interactions, and then there were end-to-end tail interactions, both London dispersion force. So we're going to see a similar thing when we talk about the structure of the double-stranded DNA. Okay, keep that in mind. Okay, nitrogenous bases. So that comes off of number 1 carbon. And the reason why we call them bases is because they're chemical bases, which are the opposite of acids. So acids give off H pluses and bases accept H pluses. And one of the most typical types of base, and we're talking about organic bases, that means found in nature, are amines, and we talked about amines in the last test. Well, when we look at the structures of our bases, we'll see that they have a lot of nitrogens in them. So that's why they're called bases, because they have a lot of nitrogens in them, which can absorb hydrogen. Now, we don't care about that in terms of their chemical properties right now. But I just want to let you know that's why they're called bases. They're not called bases like, baseball or other things, okay? It's because of the chemical properties. All right, so DNA and RNA, they both have, there's 4 bases. So DNA and RNA, all the DNA nucleotides will have their sugar and their phosphate in common, but they can have 4 different bases: adenine, guanine, cytosine, or thymine. You won't have to know the names, just know the letters A, G, C, and T. RNA is the same way. All RNA nucleotides have phosphate and ribose common. But what's different is they will have, again, there'll be 4 different types of RNAs with these 4 different bases. And DNA and RNA are very similar, So it's basically the same language. The difference is that it uses uracil, U, instead of thymine. Okay, so these are the two types of bases. There's pyrimidines and there's purines. Okay, now take a look at the structures. You can see that pyrimidines have this one ring structure, and the purines have these two ring structures. Okay, what you're going to have to know is if I showed you these guys, you won't have to memorize these structures. But if I showed you these different structures, you should be able to tell me if they're a pyrimidine or purine. Now, one way that I remember learning this is that pyrimidines have the smaller structure, and purines have the bigger structure, kind of the opposite, small, big, big, small. Might help you, I don't know. The other thing you're going to have to know is that the purines are cytosine C, T, and U. And the purines are A and G. I circled the amines, which are bases, okay, which are the base. So this is what makes this a base. Now, the other thing about these bases is actually they're pretty hydrophobic. There's a lot of carbons here, and these are big structures, and so these structures tend to be hydrophobic. Okay, they do have some hydrophilic parts with oxygens, but the big ring structures really kind of give them a really big hydrophobic character. But we're going to have look that also has hydrophilic characteristic as well, with the nitrogen and oxygen, which we'll see. So the base of a nucleotide actually kind of has two qualities. It has a very hydrophobic part, and it has a little bit of a polar part, and we'll see how that works and down the road a little bit. So what you're going to have to know here is that given a structure, you'll have to tell me if it's pyrimidine or purine, and you have to tell me if I said C, what is it, a purine or pyrimidine? Okay, so now what we're going to do, so that's the basic structure of a nucleotide. And what we're going to do now is that we're going to combine the nucleotides together to form a nucleic acid. So we're going to have nucleic acid strands, just like we did have protein strands. And protein strands were made out of amino acids. Nucleic acid strands are made out of nucleotides. So nucleic acids are a chain of nucleotides, just like a protein is a chain of amino acids. Okay, the phosphate group of each nucleotide, we're going to learn in a minute, forms a covalent bond to the pentose of the adjacent sugar. So see down here, so what we have here is we have 4 nucleotides, and they're all linked together. And we'll talk about the link in a minute. But so here's number 1, here's number 2, number 3, number 4, and what happens is the phosphate of number 1 connects with the number 3 of number 2. Okay, and so what we're going to do in a second, we're going to find that now there's another role for another carbon. The first carbon bonds the base. The second carbon tells me if I have RNA or DNA. Number 3 now, number 3 carbon bonds to the number 5 carbon of the previous nucleotide. Okay, number 4 carbon. All right, now this 5 should actually be up here at this end right here. The number 4 carbon, it's just part of the structure. It's needed. It doesn't really have a role, but it has a role in holding the structure together. But the number 5 carbon connects to the phosphate, and therefore all it connects to the number 3 carbon of the next nucleotide. So what we have here is number 1 binds to the base. Number 2 tells me if it's RNA. Number 3 binds to previous nucleotide. Number 5 binds to the next nucleotide. Number 4 is just part of the structure. Okay. All right. So how does this happen? So what you do is you start out with a single nucleotide. And what you do is then you take another nucleotide, and that nucleotide is going to fall on top of the previous or original nucleotide. So this number 3 is going to connect what we say to number 5. There's a phosphate cell coming off number 5. And what it does is it forms this bond right here. So what happens is there's a 3 to 5 phosphodiester bond. So it's a 3 to 5 bond. Now you don't have to name this, okay? You might be on like a multiple choice problem or something like that, but the name tells you the direction. The 3 bonds to the 5. It's not the other way around. So the 3 comes in and bonds to the 5. That's important because you have to have this guy first before you have the other. So it's the 5 doesn't go to 3, so you have to have this guy first, and then the 3 will bind to the 5. Okay, and when you do that, now you have basically a starting of a nucleic acid. Okay, where you have this nucleotide and this nucleotide. Now they're exactly the same, except the bases, okay? Well, the bases could be the same or different. So this is the variable part. Now, typically when I'm in class, I ask you guys, I say, is this RNA or DNA? And so if you remember, the way to tell, there's 2 different ways to tell if you're RNA or DNA. DNA, RNA has an oxygen coming off of number 2. Okay, so this must be DNA because there's no oxygen coming off of number 2. So this is DNA. And then I would ask, is this a pyrimidine or purine? Okay, it's a bigger structure, so it's smaller name, purine. Is this a pyrimidine or purine? This is a pyrimidine. And then you have to ask is, if this is a purine, which one is it? Well, you don't know. You don't have enough information. Okay. So we're going to expand the idea and grow on it. So you had nucleic acid, nucleotide 1, nucleotide 2. And what they did is they connected the phosphate to the 3. And so now you have your beginning of your nucleic acid. And so this is your number 1. And if you notice, number 1 has a 3 on the end. And now what you're doing is number 2 has a 5 on the end. And here's your phosphate I asked about. And then what you do is you keep on adding. So next one, you add here. You don't add it down here on this end. You don't add on this 3' end, we say. You always add on the 5' end. Okay? Since like amino acids, what you did is you always added on the carbon. All right? You didn't add on the nitrogen end. So this has direction, just like the amino acid sequence of a protein. So the very first guy will have a 3 that's not connected to anything. So the next one comes in, and a 3 connects to 5, and a 3 connects to 5, 3 connects to 5, 3 connects to 5, 3 connects to 5. Okay, and now what we have here is a 1, 2, 3, 4, 5, 6 nucleic acid. Okay, and so these parts are all identical. Now I don't know if this is RNA or DNA, but it doesn't matter. But all these parts, so these rectangles represent the the sugars, and then here's the phosphates. These are all identical. What's different, or may be different, no, they don't have to be, but are the bases. Okay, so you'll have, at the very beginning, you'll have an open 3. We call it free 3 prime. And at the very end, you'll have a free 5 prime. So what we say is the chain grows from 3 to 5 prime. Okay. All right, so let's take a look at that. So this is a nucleic acid that has 4 nucleotides. This is number 1, number 2, number 3, number 4, and number 4. Now, how do I know this is number 1? Okay, well, because the number 3 is free, and how do I know this is N? Because it has a phosphate coming off of it. Okay, so which is the 5 prime. So the number 5 prime. So when the 5 prime is the end, so the beginning, so it goes in this direction. Number 1, number 2, number 3, number 4. And so here are my phosphodiester bonds. So 3 to 5, 3 to 5, 3 to 5. Number 1, number 2, number 3, number 4. Okay. Now, okay. So what we could do is name this nucleotide, this nucleic acid, according to the bases, because that's what makes them different. Okay. So this particular nucleic acid, we could say, is a uracil, guanine, cytosine, adenine. So we could write down UGCA, and instead of writing or drawing this whole structure, and that would imply all this structure. So if I wrote down UGCA, and I knew all these chemical formulas, I could write down all this stuff, and I would have what I need. I would actually start out, I'd write down 3 prime UGCA 5 prime, and I might go left to right or up, down, whatever, because it has a direction, like I have over here, 1, 2, 3, 4. Now the other thing, so again, in my class, what I'll ask once I put this up on the screen is I'll ask if this is RNA or DNA, and I'll ask why. And people go, "Oh, it's RNA." And I go, "Why?" Well, there's 2 ways you can tell. One is the second carbon has oxygen on it, so it's a ribose. The other way to tell, it has a uracil instead of a thymine. Okay. All right, so this is an RNA. Okay, so here's a kind of a cartoon. Let's not draw the whole structure. Let's write it out in a kind of way that gives the same information, but it really accentuates what we need to know. So we have our phosphates out here. We have our sugars. Notice there are five rings, the pentosan. And then we have our bases, okay? And all these could be all the same, all different. This could be G, 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 C, 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 A, C, T, G, or whatever. And that's what's going to be important. Just like amino acids, their amino acid sequence was important in their order. Nucleotide sequence is going to be important as well. So this is number 1 nucleotide, this is number 2, this is number 3, and this is number 4. Okay, so this guy right here, this is nucleotide number 1, and it combines 3 to 5. Okay. All right. Now, the other thing to notice about this is this RNA or DNA? Okay. Well, the way to tell is that, I mean, you can't tell because the sugar here, because it doesn't give enough information. But if you look at the bases, the bases have A, C, G, and T. If it was RNA, this would be U. So this would be a DNA. Now notice the other thing about this, the way it's drawn, is it looks like you have some complements here. It looks like the T could kind of complement or fit into the A, and the G and the C could fit into each other. Okay? All right, the other thing is which ones are pyrimidines and which ones are purines. Okay, so the pyrimidines we said are the smaller structures, so those are the C and the T, and the purines are the big structures, the A and G. Okay, so just like proteins, nucleic acids aren't just long chains of nucleotides. Okay, they're not just extended long chains. They have three-dimensional structure. They fold up. They end up having very unique structures, just like proteins, okay? And have very similar reasons as proteins do. And phospholipid bilayers, and soap and oil. Okay, so phosphates are polar. So that means they'll interact with water. The bases are nonpolar, and they will try to get away from water. Now, I went through explaining why this happens. That I said that water gently pushes the nonpolar parts so they get with other waters and stuff like that. But right now we'll just say that they'll try to get away from water, and that's fine. And so, but we also, that's because they have a lot of hydrophobic capacity, which I showed on a previous slide. Those ring structures are very hydrophobic. But the other thing is that the bases can form hydrogen bonds with other bases. That's going to be pretty important. Now, one of the things that RNA does, it's a single chain, and what it does is it folds up just like a protein. And here's kind of an example of it. So this is just one chain of RNA, and it folds up, okay? And it folds up for kind of the same way as protein. So the nonpolar parts, which are the bases, they kind of go to the middle, and then then there's polar parts on the bases that will interact and form hydrogen bonds, and that's what these little connections are here. And then on the outside are phosphates. And so proteins kind of fold up. So RNAs kind of fold up like proteins for the same reasons. And it's a single chain. Now DNA usually, it can have double chain DNA, but single. So it's usually thought as a single chain. Now DNA, and everybody's probably seen it, has two chains that make it up. So a double chain, and both of them do this helix type thing. So this is one chain right here, And this is another chain. So DNA, when you look at it, it's actually two molecules, not one molecule, that are interacted and have very specific interactions, which we're going to talk about here. So let's look at RNA's three-dimensional structure. So this is kind of like a tertiary structure, okay? So it had a nucleotide sequence, and that nucleotide sequence actually told this structure how to form, just like in proteins, okay? I mean, nature tends to repeat itself over and over and over, okay? So, so when you look at this structure, there'll be phosphates hanging on the outside, and they'll form dipole with the phosphates. There'll be hydrogens, hydrogen bonds, forming between the ends of the bases, and there'll be London dispersion forces happening between the sides of bases, and we'll have this will be called base pairing, and this will be called base stacking. Okay. Now, when we had phospholipid bilayers, what you had is you had a polar head, and then you had your polar tails. And what happened was the one phospholipid would interact with another one, and so they would interact end to end, tail to tail. Okay, but what would happen also is the phospholipids would interact side to side. Okay, so the tails would interact side to side. Again, through London dispersion forces, and then you had the heads would interact with the water. Well, you get kind of the same thing here. You'll have the tails interacting end to end, and then the tails also interacting side to side, and the heads interacting with water. The difference is that instead of having the ends interact through London dispersion forces like in phospholipid bilayers, they interact through hydrogen bonds, and that's way more stable, and it locks in the structure here. Okay, and we'll see how that works again in DNA in a minute. And I'll count down. 54, 55. Okay. All right, let's talk about DNA. All right, before I continue on to deal with DNA, we must mention differences between RNA and DNA. RNA is usually a single strand. It doesn't have a double helix structure that we're going to talk about in a second. It folds up like a protein. The other thing about RNA is, there's two things about it. It has a uracil instead of thymine, and it doesn't have, it has an oxygen. Now, what we're going to find is that in DNA, thymine will hydrobond with the adenine, or a T to an A. For RNA, it's going to be a U to an A. Okay. If you can, try to remember that, and we'll see that specifically when we talk about forming proteins. Okay, so there's three types of interactions in when an RNA or DNA form. One type is phosphate with the water, the polar ends interacting, but the other part is how the bases interact with each other. So the bases get buried away from the water. So if you look at RNA or DNA, the interior of RNA or DNA are where the bases are. Okay, and what they do is they can have two different kinds of interactions. They can have interactions end to end. So over here would be one nucleotide, and over here would be another nucleotide. And specifically, what happens is where they form hydrogen bonds, which we told you is a special kind of dipole-dipole, like an alpha helix and beta sheets. Now, G's will always form hydrogen bonds with C's, and A's will always form hydrogen bonds with T's, okay? And if you look at them, you can see the difference. And G's with U's and A's with U's. So you can see A, G, G, U. Okay. All right. They can form these. This one. So these. So GC, AT, or G. So this one can happen, but not as much. So the ones we'll see most of the time are GCs, ATs, or AUs. This one can happen, but we don't see it. It kind of gives you a different kind of structure. But it can happen in like RNA. But we'll see in this in RNA and DNA. But so anyways, so these dotted lines are hydrogen bonds, okay? And they happen end to end. In phospholipid bilayers, it's like the phospholipid bilayers where they had end-to-end London dispersion forces. The end-to-ends are hydrogen bonding, which gives it way more structure and holds it into place way better. The other type of interaction is the bases can stack up. We'll see in a minute, but they stack up on each other, and these are London dispersion forces. Okay, so this guy over here, the green, is a base, and you can see the ring structure. That ring structure forms London dispersion forces with the guy down here, and it's really quite stabilizing. Okay. Okay, so we saw, side-to-side interactions with phospholipid bilayers. We also have side-to-side inside the RNA or DNA where the bases stack on each other. And these are very strong London dispersion forces. And here's different examples of it. So you can see it here and here and here. Okay. And it's a very important thing. Okay, I remember when I first heard about it back 30 years ago. I was just amazed when I heard about it. Okay. All right, so now we're going to look at DNA. So RNA, what it does is it's a single strand. It folds up. What happens is nonpolar parts go to the middle, the bases. And then the bases, when they go in the middle, they will form the base stack, London dispersion forces, and they'll also form hydrogen bonds between the bases. Okay. Okay, so now we're going to talk about DNA. Now DNA, and specifically double-stranded DNA. Okay, so now we're going to look at it. So DNA is of an organism may contain from several thousand nucleotides to a billion. The DNA from different organisms may differ in their length and sequence. So basically, is the DNA carries the information of who we are and what we're going to be. Okay, it tells us how to make our proteins. Who are our proteins, and our proteins will tell us how to do our cells to do stuff. Okay, so this, and it's information. Okay, it's basically information, not really a job. Okay, it's just kind of like a storage of information. The proteins do the job. These guys store that information. All right, we've probably all seen this beautiful structure of DNA. Okay, we just had this past week, April 25th is called DNA Day, and that's the day that Watson and Crick put out their famous paper describing the structure. And the story of how they came up with the structure is really, really, really amazing. Okay, it's incredible to think about how they came up with it, given the tools that they came up with it and the information they came up with it. Now, a side note is that Francis Crick and James Watson, um, they actually didn't. They had no desire, never thought about figuring out the structure of DNA. They met each other in a pub in England while they were both doing research, not on DNA, on different things. Watson was doing DNA. He was doing research on plants, and Crick was doing research on X-rays or something. Okay, neither of them had any interest in biology at all. Well, they kind of found each other at a pub, and they started talking about this, and they decided to go, well, let's see if we can do it. So it wasn't part of their day job. And at night they got together and they finally came up with it. There was a TV movie a few years, 20 years that came out. Now, the only reason why anyone would be interested in watching it is not because it's about DNA, it's because who's in it. It has Jeff Goldblum, okay. And I know Jeff Goldblum has a huge following these days. But so this was like 1991. So it's kind of a cool movie. It's called The Double Helix. If any of you are interested and you're sitting home during coronavirus and you have nothing to do, if you're sick of watching The Tiger King, go look up Double Helix. It might be kind of fun. But anyways, so what you have here is the idealized structure of DNA. It's double-stranded. That means there's two chains. Okay, there's one chain going like this, coiling up, and there's another one coiling the other way. And actually, the two coils go opposite of each other. So there's one coil going up, and there's another coil going down. All right. The other thing to notice is that red means oxygen, blue means nitrogen, white means hydrogen, and black means carbon. All right. And their phosphate is also blue. All right. Now, if you notice, you probably don't see a whole lot of carbon. Right. Now, the most abundant atom in DNA is carbon. So where is it? Where is it here? Okay. Well, from the last test, we learned, and the previous test, is that a molecule with a lot of carbons tends to be nonpolar, hydrophobic, and tends to stay away from water. So this picture here is not a model just sitting out in space. It's supposed to be a model of DNA in water. So you see this blue, this is supposed to be water. And so you have the oxygens, the reds, are forming dipoles with water. Okay. And also the blues, the phosphates and nitrogens, form dipoles with water. And the carbons, well, they kind of hide away. Okay, for the reasons that we all just talked about. Okay. So we're going to get into the structure more. So you have two strands. Okay, and you have polar and nonpolar parts. Polar parts on the outside, nonpolar parts in the middle. The oxygens are from the phosphates. Phosphates have 4 oxygens on them. Okay, so if we look at the nucleotide structure, like I've been saying all along, it has, so we're going to compare it to soap and a phospholipid bilayer. So soap has a polar head and nonpolar tail. Phospholipids had a polar head and two tails, nonpolar tails. A nucleus structure is very similar. It has a phosphate, a sugar here, which is neither or, and then it has a nonpolar base, but it has possible parts can do form hydrogen bonds. Okay, so remember proteins. Okay, in proteins, when they folded up, sometimes polar amino acids got stuck inside the middle. And when they got stuck inside the middle, they would form hydrogen bonds. Well, that's kind of what's going to happen here. So what we're going to find is just so when you have phospholipid bilayer, what happens is they had another phospholipid bilayer over here, and another one over here and over here. And so there will be side-to-side interactions here, and then there will be end-to-end, and they were both London dispersion forces. Then you had your dipole-dipole. For soap, you just had you had end-to-ends, maybe some side-to-side non-polars, and then you had your polars. For proteins, you had your polar amino acids on the outside, your non-polar in the middle, and every once in a while you had a polar guy in the middle that would find another polar guy in the middle and form a hydrogen bond. Well, DNA is going to kind of combine all this stuff together now. So you're going to have the polar phosphate, which is like a head, and nonpolar. And so this is going to stick on the outside. This is going to stick in the middle and interact with the other nonpolar bases. And it's going to form base stacking. And then these polar parts are going to find each other and form base pairs. And here you go. So this is the big picture of how all this sticks together here. So you have two chains. So this is our chain going 1, 2, 3, 4. You have your 3' at the end and your 5' up here. And this is DNA, has a T in it. So TGCA. The phosphates stick out and they interact with water. The bases stick in the middle because they're hydrophobic, and what they do is they form base stacking, right? Like in previous diagram, okay. And these are the hydrophobic interactions with London dispersion forces. Now, the bases also have polar parts that will interact with each other through hydrogen bonds. So specifically A with T, a C with G, and so forth. So this is how the two structures go. So you have one strand going up and one strand going down. And specifically we'll see that whenever there's an A over here, there'll be a T connecting to it. You can see it kind of fits right into it. A C to a G and so forth. Okay. So that's the general structure of DNA. Whether you'll take it with you somewhere and ever talk about it ever again, I don't know. I just think it's cool. All right. Okay, so again, inside, so this is a DNA. So it has a beginning 3' to 5', and then another guy going 3' to 3'. So it goes, one goes one way, this one goes this way. And there can be hydrogen bonds between A's and T's and G's and C's. Okay, just like proteins, the polar parts of the bases get stuck in the middle and they form hydrogen bonds. And the hydrogen bonds will specifically form usually between A's and T's and G's and C's. So usually on one strand, if there's an A, on the other strand, there will be a T, and we'll call that a complement. So the complement of A is T, and the complement of G is C. So the two strands run in opposite direction of each other, so one goes up, one goes down. They're not identical. Okay, what they are is complement. So if the up strand goes the up strand goes A, the down strand will go T. Okay, so you have a primary strand, and then you have its complement. All right, and here's an example of it. So here's your primary. It's C. It's 5 prime. Okay, actually, it's the other way around. This should be the primary strand up here, and this should be the primary complement down here. But anyways, so you have S stands for sugar, P stands for phosphate, and this is G, which is guanine, thymine, C, and it doesn't have to be this order. I just picked it: A, G, C, T, and C. So let's start here. Okay, so this is my first one, C. So this is my first nucleotide, has cytosine. Second one, adenine. Third one, thymine, and fourth one, guanine. All right. So on the other strand, you will have its complement. G will hydrogen bond with C. T will hydrogen bond with A with T, and C with G. So a simple way to do it is, instead of drawing all this out, because these are constant, the backbone, we call this, is constant, so we're not going to write it down all the time, is that all we have to write down the differences. So we write down the differences. So this is 3' GTAC, 5', and the bottom strand is 5' CATG, 3'. Now if I saw this and I knew everything about the structures, I could draw this out. I go, oh, I know what C looks like. I could draw that out. I know what G is. Draw it out. And I know how they connect. Boom, boom. Okay? So this is a shortcut. Okay? So whenever you see a C, so the reading goes CATG, its complement would be GTAC. All right, so here's a little practice. So what I want to do is, I have two strands here. Again, I have them backwards. It should be 3' to 5'. So what I want to do is find the complement of these. All right. So wherever you find an A, there will be a T. Wherever you find a G, a C, and so forth. Okay. So I'll let you try to figure that one out. You can turn off the turn it off and try to figure it out. Next slide's going to have the answer. All right. So this is what you have now. So you have so this is your primary, TTAG, GCATTTAGC. The complement is AATCCG, TAAATCG. And the same goes here. All right, so this is a good question to ask on any test, is if you given a primary sequence, what is the complement? Okay. All right. Just like with proteins, carbohydrates, and lipids, what we did was we covered structure first, and then we said structure implies function. So one of the questions on the last test was a fill in the blank. It said this unique causes unique causes unique. Okay, let's go back to that one because it's really important and it relates to this. So a protein, the amino acid sequence is called the primary structure, and it gives the information for the protein to fold. So the amino acid sequence provides information. So a unique amino acid sequence gives a unique protein structure or tertiary structure. Okay. the structure of anything determines its function. So if you know its structure, you know its function. So it was sequence determines structure, structure determines function. That's been the whole story for the whole semester. Day one, if you go back to the very first lecture, I think I said that. Said structure determines function. And I've talked about, Corvettes and semis, okay? Semi trucks is that, and trees and all kinds of things, okay? Is if you know the structure of something, you pretty much can figure out the function. So same thing goes here. What we always have done is we study the structure first, and from the structure we hope we can figure out the function. Okay, so now that we've gone over the structure of DNA, we studied the structure of nucleotides, and then we combined the nucleotides together, formed nucleic acids, and came up with structures of RNA and DNA. So now what we want to do is, how do those structures relate to how they function? And primarily what we're going to do is talk about how the structure of DNA relates to what it does functionally. There are going to be two things we're going to be looking at. We're going to look at something called DNA replication. That means, how does DNA make more of itself? Because you need to do that. And then how does DNA do what's really important? Make protein. So we're going to talk about DNA replication. So in your cells, if you've had biology, and I hope you have had it, A&P, I hope even a basic biology class, as you know, is that cells are constantly replicating. Your skin cells are constantly dying, and what you need to do is you need to replenish all the cells that are dying in your body. Okay, now when you replenish your cells, you just don't replenish the cells. You have to replenish the information of the old cells. You have to have the information that they have move on to new cells. So what your body does is, and there's two different ways, two different reasons it makes it. One thing is that what's happening all the time in our body, like I said, we're having cells dying all the time, and there's new cells being made, and each cell that's being made has to be exactly the same as the other. What I mean by exactly the same, it's got to have the same proteins in it to do the same jobs. That means it has to have the same DNA, because the DNA determines what the proteins is. So when a new cell is made, the old cell will make identical copies of its DNA in the new cells. And that's called DNA replication. Okay. All right. So let's say I have a cell, a skin cell, and it's going to replicate, get new cells, new skin cells. So what I want to do is replicate the cell and replicate the DNA in the cell, because that's really an important part on it. DNA is in the nucleus, at least in eukaryotes. Again, and we won't go into this right now. All right, but so we're talking about cell division, and maybe you learn about this in biology, but there are two kinds of cell division with meiosis, and that's when the sex cells replicate, and it actually happens twice. It does a double kind of division, and then you have mitosis, which are your like your body cells, like your skin cells, and how they replicate. But what we're going to look at is how the DNA in your cells is replicated. Okay. So every time a cell, actually, what's pretty cool is every time a cell makes a cell, it doesn't just make one cell, it makes two cells. All right. So it's kind of, which is kind of cool because maybe one of the cells is wrong and die or something, and the other one is good, or makes two good ones. All right. So so it's kind of cool that it makes two out of one. So let's go on and see how that is. So this is gonna be a really simple version of it. You know, when you, I don't know, if you learned about this stuff before, you learned about what's anaphase, prophase, metaphase, interphase. Okay, and you know, you saw the cool looking pictures that look like they're magnets and things like that. We're not going to get into that. We're just kind of talk what happens to DNA. All right, so what you do is you start out with your, what we'll say, our mother DNA, our original cell, and we'll say its DNA is TACG and the complement is ATCGC. Okay, and these dashes are hydrogen bonds and base stacking, and the dark bonds are actually the covalent bonds between the nucleotides. Right. So the first step, and we won't talk about how all this gets started, but the first step is that these two strands have to be split apart from each other. And so there's this enzyme called DNA helicase, and what it does is we say it unwinds or unzips the two strands from each other. Basically, it just splits them apart. Okay, so you can see this is the enzyme that's kind of going here and splitting apart the two strands. So now you have two single strands of DNA. So you started with a double-stranded DNA, and you ended up with two single-stranded DNAs. Okay, next thing that happens is so the hydrogen bonds are broken. That's what we just talked about. So now you have two single strands instead of one double strand. So that's what the helicase did here. All right, so now you're all ready. Next step is, well, okay, so there's a pool of nucleotides, we say. That means just a lot of G's, the T's, A's, the C's floating around. And what they're going to do is kind of come up and connect through hydrogen bonds to the just each strand. Okay, so A will hit to a T, A, T to an A, G and C, and so forth. And they're going to form a hydrogen bond. Well, that's all and good, but that doesn't. You haven't made a connection. You haven't made a strand yet, because these guys are not connected with each other. Okay, so they're just sitting out here wiggling around, connected to the T. Okay, but they're not all connected to each other. Okay, and the same thing down here, and that's what the next. So the next step is this enzyme called DNA polymerase. Polymerase means it makes long chains of DNAs. And what it does is it snaps together the bases. Okay, so what it does is these guys that are dangling off now form covalent bonds with each other. So now you have, this is a strand here, and strand. So the circle part is your original strand, and these are your new strands here. And you're done. Okay, and when you're done, what you have is you have two identical DNA, double-stranded DNAs to the mother DNA. So we call these daughters, okay? And if you notice, the mother starts at TACG, ATGC. The daughters end up being exactly the same, okay? So now you can add this go to one cell, and this would go to the other cell. Okay, and this happens in meiosis and mitosis. All right, the next function of DNA is going to be how DNA is involved with making more protein. This is called protein synthesis. Okay, so any protein. So every protein is different. So if every so a protein, okay, it has to be made. And how the information to make a protein comes from DNA. So there's DNA that codes for protein. Every protein has its own DNA. And we call that DNA a gene. So every protein has a gene. Every protein has a unique amino acid sequence, and that's because every gene has a unique nucleotide sequence. So the gene, so going back to last test, said the amino acid sequence determines structure, structure determines function. We can add another layer here. The DNA sequence, the unique DNA, unique nucleotide sequence, makes a unique amino acid sequence, which makes a unique structure, which makes a unique function. So a gene, and everybody's heard of gene, is a gene is a part of DNA that gives the information to make a particular protein. So hemoglobin, the protein in our blood cells that carry oxygen, has a unique amino acid sequence. And what is that unique amino acid sequence? It comes from information of a gene, a DNA molecule that is specific, that gives the information to make the amino acid sequence. Humans have about 20,000 genes. That means we have about 20,000 different proteins in our body. Okay, it's pretty amazing. Now, and so there's 20,000 different genes, and we make probably 500, 1,000 of each of these every time we make them. So there's probably over 2 million proteins floating around in our body at any given time. Okay, that's pretty crazy, all that stuff that's going on. All right, and every cell, it's really cool, is every cell has all the information that every other cell has. So your liver cell has the information to make heart cells. Your brain cells have the information to make liver cells. But what's kind of cool is your brain doesn't make liver cells. So, but it has all that information. It's a crazy world. It's with DNA. Okay. All right. So let's go on here. So in protein synthesis, which we mean, what is involved to make a protein? There's going to be two steps. The first step is called transcription. Second step is called translation. The first step is you're going to take DNA and you're going to make RNA. And the reason you're going to do that is you want to keep the DNA preserved. It's special. So you don't want to just send it out and use it all up. What you like to do is keep a central copy of it. And so, because it has all the information, you like to reuse that information. So what you're going to do is that you're going to make copies of it, and you're going to send it out. So, so DNA is like your blueprint uh, of of your information. So what you do is you're going to need to make a copy of that blueprint, and that's going to be the RNA. And we call it transcription because the copy of the from DNA and RNA is basically just kind of you're going. It's the same language. You're going to be changing the language of DNA to RNA, which are basically the same. The only difference is going to be for DNA you're going to have a T, and RNA you're going to have U. So basically it's the same language. So you're basically, I don't know if you've transcribed anything. When you transcribe something, it means you're basically trying to write word for word for something. So this is sort of word for word. And then the second step is called translation. In translation, what you're going to do is you're going to change languages. You're going to take RNA language, which is nucleotide, and then you're going to change it to a whole new language called amino acids. Okay, so the process by which genetic information of an mRNA molecule specifies the amino acids. Okay, so the mRNA is going to be translated, in a sense, decoded. So this is like a computer program. So what you're doing is you're going to be decoding. So in the first step, you're just sort of rewriting the code. In the second one, you're actually decoding from RNA to amino acids. So it's RNA to amino, which is a different language, just translation. So it's like English to French, different language. You're going nucleotides to amino acids. All right, so I'm just going to write it down here, okay? I could draw pictures and have all kinds. There's all kinds of videos on YouTube on this, but I'm just going to go ahead and tell you how it works, and hopefully you can see it kind of in your head. I would suggest you, if I maybe in the next couple of days, I'll find some good YouTube videos to that you can use kind of reference this. But you have two steps. You have transcription and translation. So the first step is to have the information of DNA turn into RNA, and it happens in the nucleus. Okay, so what you do is you take one of the DNA strands. So what you do is just like in DNA replication, you have unzip it. And you don't have to be specific about that, but you take one of the strands, and that strand is copied just like in DNA replication. Well, you get a complement. So if you have, you have your sequence, you make a complement copy of that. So if you have like a CCC, you'll get a GGG, and we'll get into that in a minute. So one of the strands is a complement copy is made into RNA, and that's called a messenger RNA, which you'll see why it's called messenger in a minute, or mRNA. And the protein that does all that stuff is called RNA polymerase. Now, in DNA replication, it was DNA polymerase that made DNA to DNA. This case, you're going from DNA to RNA, so it's RNA that makes the RNA. So after the mRNA is made, there's some steps that need to be made. It's edited. Some parts need to be removed. So it's for different reasons, to protect it, and also some parts don't really need to be there. All right, after it's been edited, the mRNA now will leave the nucleus. Okay, and it'll go out into the cytoplasm. Okay, and so what it does now is that there's this organelle called the ribosome. Should be ribosomal complex, or just RC. And again, you might have seen, like if you've taken biology before, you might have heard of, your smooth and rough endoplasmic reticulum. The difference between the smooth and the rough is rough will have the ribosomal complexes on them. But anyways, so what happens is ribosomal complexes are kind of like a factory. And this is the place where the proteins are going to be made. And so what happens is the mRNA enters the ribosomal complex, so it binds. And when it comes in, when the mRNA comes into the ribosomal complex, so you have this RNA that has, it's a long chain of nucleotides like AA, TTT, whatever, all the way through. Is the ribosome complex. It reads the first three nucleotides it sees. Okay, and a set of three is called a codon. Right, and so it reads those three. Now I'm going to oversimplify this and kind of make it like in terms of humans, like factories and all that. So after reading the codon, the three, what it does is it calls out. So it may read a AAA. It says, what it does is it calls out. It says AAA, and there will be these guys called tRNAs, which will respond. Okay, so they'll hear the ribosomal complex called out AAA, and then the guy that understands the AA or responds to the AA, because that's who relates to it, okay, will hear it. Okay, so the AAA tRNA will come and attached to that AA tRNA will be a specific amino acid. We'll get to that in a second. So there are 64 tRNAs, and those 64 carry the 20. So that means that each amino acid has will have more than one tRNA that's specified for it. So it calls out for the TNA, so AAA, okay, and the AAA tRNA hears it and says, okay, I'm going to bring down my guy. And so it brings down the associated amino acid that's associated with AAA, and it drops it off. So it leaves it with the ribosomal complex. Then the ribosomal complex does next is it moves the whole RNA over 3. Okay, so you have this long chain, okay, and so it read the first 3, called AAA, brought down AAA amino acid, and right now it moves over 3. It reads the next 3, calls out its specific tRNA, finds that has a specific amino acid, brings it down. Okay, now what it does is it connects that amino acid with the preview amino acid, and they form the beginnings of a polypeptide molecule. Okay, so it keeps on doing that. So it shifts over 3 again, reads those 3s, finds the appropriate tRNA, brings it down, attaches it, shifts over 3, so forth. Then finally, there's a stop codon that has like UAG, and we'll see this in a minute. and when it reads a stop codon, it says, okay, we're all done. And when it's all done, it says, just stop reading and let the amino acid chain leave. And so now this amino acid chain has several amino acids all linked together, will leave the ribosomal complex, and they will go fold in its tertiary structure and go do its job. Okay, so this is kind of a picture of what all goes on. So first thing, you have the mRNA is made by the DNA, and then it leaves. Okay, and then the RNA bonds to the ribosomal complex, and calls, and these are tRNAs. They have amino acids. They come in a link. They drop off the amino acids. Move over 3, bring a new one in, connect it, and then you have your chain. And then when you hit your stop codon, this guy falls off and it goes folds. All right, now let's talk about the tRNAs. Okay, so there are 64 different tRNAs, and those 64 will carry the 20 amino acids. Now, there is a 1-to-1 ratio, so the 64 don't carry 64. That means that some of the tRNAs will carry, some of the amino acids will be on more than one of the tRNAs. We'll see in a second. So each tRNA has a specific 3-letter codon. So codon is 3 letters. On the next slide, we'll see the 64 tRNAs and their corresponding amino acids. Okay, so let's just go through an example. And you may want to page up to the table, genetic code table, to see how this works. But I'll lay it out here. So let's say the ribosomal complex reads the mRNA and it reads the AAA codon. Right, and then what it does is basically it calls out for a tRNA that has an AAA that recognizes AAA. On that tRNA that has an AAA with it, it also has the amino acid lysine attached to it. Okay, so it's called out for and brought to the RC. So when it reads this AA, it'll bring back an AAA. The tRNA will come down, and with it, it'll bring a lyse. Okay, so now it'll move over 3, move over to G, and now it reads the G, and it says, go find me the GGG tRNA. Well, the GGG tRNA has a gly attached to it. Okay, and what it'll do is it'll bring down the RC and drop the gly off, and the gly will attach to the lyse, and you have a lyse-gly a chain forming. Okay, then I'll read the next 3. So in the next table, we'll see how this AA relates to lyse and the GGG relates to gly and so forth. Right, so this is it. This is called the genetic code. All right, so this is using RNA. So instead of T's, you'll find U's. Okay, so in the previous example, I said you had an AAA, and notice it's right here. AAA is a lysine. So there is a tRNA called AAA, and it carries a lysine, or LIC. But there's also another codon called an AAG tRNA that also called a lysine. So it's a way of having, more opportunity, more options by having more chances, and it's really cool why all this stuff is, and I mean it's not random. Why, like, leucine has 1, 2, 3, 4 different tRNAs or actually six, 1, 2, 3, 4, 5, 6 different tRNAs that will code for it. Okay, uh, there's reason for all this stuff. I mean, and so this table is really interesting in terms of its site. But I talked about stop codons. So on on the mRNA, there will be codons either UAA, UAG, or UGA that don't have tRNAs for them. So when the ribosomal complex, the RCs hit a UAA, UAG, or UGA, it says, okay, we're done. And the protein falls off and goes and folds, and we're all done. Okay, so what we're going to do is use this table, and on tests you'll be given this table, have it available to you, and you're going to have to use it. So you'll be given like a UUG, what is the appropriate amino acid that's related to it. Right, so here's an example of going from transcription and translation. So in terms of the actual information. So let's say that our DNA is TTT, AAA, GGG, CCC, TTT. So it's DNA, right? So we're just going to take this one strand. We don't care about the other strand. So the first thing that happens is the RNA polymerase is going to make a complement RNA. Okay, so wherever T, it makes an A. Wherever A, it makes a U. Now, if there's a DNA, complement DNA, it would be a T. So it's AAA, UUU, CCC, GGAA. Right, now this guy here goes out into the cell, into the ribosome, and it connects to the ribosome. And the ribosome translates this into amino acids. So what it does is reads the AAA first, calls out for the appropriate tRNA, an AA tRNA, and on that AAA tRNA there's a lysine. It brings back, puts the lysine on the RC, and leaves it there. Okay, then it shifts over and reads UUU, calls out for the tRNA, UUU tRNA. Attached to that will be a Phe, phenylalanine. Or bring that Phe, leave it on RC, these two guys connect, and moves over 3. Then it reads the CCC, finds the tRNA, that's CCC, that tRNA has proline attached to it. Go back and look at the genetic code. And brings it down, leaves the proline, and it connects to the Phe. So you have lysine, phenylalanine, and proline. Again, what it does, it goes up, reads, then what it does is read GGG, finds the tRNA, GGG, which has glycine, brings the glycine down, drops it off, connects it. Now you have lysine, phenylalanine, proline, and glycine. And finally, and I didn't have a stop codon here, but it'll go down and do AA again, and so forth. Okay, so what you're going to have to do on the test is, given the DNA, you're going to have to figure out the mRNA complementary structure, and from that, what is the amino acid sequence that it translates to. So the first step is transcription, and this step is translation. Okay, so that does it for nucleic acid function. What we're going to do now is just talk a little bit about some of the things that can happen with like wrong reading. So you have, as you're doing all this, you're reading, the RC is reading all these letters, and the DNA is being into an RNA, and we just go, oh, it does that perfectly. Well, in your life is if you ever try to copy something word for word, a lot of times you always leave out a word or a letter or something like that. It happens in with this as well. And so the translation and transcription is never perfect. And if you get a bad, if there is a reading, so what we have is that DNA sequence determines amino acid sequence, it determines structure, which determines function. If something goes wrong in the reading, the copying of the DNA, therefore there will be something wrong in the copying in the making of the amino acid sequence. And if something's wrong with the amino acid sequence, there's something wrong with the structure. And if there's something wrong with the structure, there's something wrong with the function. So if there's anything wrong that's important in the copying of DNA, okay, it can affect how an organism lives. Okay, and this is what we're going to talk about now. So if there's something wrong with the process of the copying, what we get is something called mutations. And mutations are when you get a change in the nucleotide sequence. And it could be because there is a misreading or whatever. We'll talk about that in a minute. And if there's a mutation, what that means is, so now the DNA sequence is not the same as original, that may not give you the same protein that you started out with, and the same structural protein. It'll be almost exactly the same, but maybe it's not. Maybe it's been changed just enough to it can't do its job anymore. Okay, here it says most mutation are harmful implications in Denver. Okay, actually, this is wrong. Okay, we have mutations happening all the time in our body, and most mutations are not harmful. And this is all. I took this from the guy previous, so it makes it sound like mutations help us survive better. Mutations are not made to help us become better. Okay, mutations happen. And if they happen and it's in the right circumstances, they allow us to be better, live better. That's fine, but the mutations are not made to make us better. All right. So this is all wrong here. I always love wrong statements. Okay. So mutations happen all the time. There's always misreadings and so forth of DNA, of translations and transcriptions, and most of them are not harmful, especially somatic cells, mitosis. There's a lot of misreads in your skin cells, stuff like, and usually they're just kind of thrown away or hardly do anything at all. Okay, so, but sometimes, every once in a while, a mutation can be a disastrous thing, and yeah, well, it can lead to things like cancer and genetic diseases. So this says bacteria mutate response to stress, like, so we do the same thing. No, we don't mutate in response. Okay, that's again, this is a wrong statement. Is that bacteria don't mutate in response? It's that bacteria mutate. All right, they just mutate. Period. And as they mutate, some of the mutations will survive better stresses. Okay, so bacteria doesn't say, "Oh, I want to mutate." All right, it says, "Oh, I'm sorry, I made a bad guy. Oh, I made a good guy." It's an accident. Mutations are accidents. Okay, they don't have purpose. And so, anyways, so what do you have to learn about this? What does this mean? So mutations are just changes in the DNA sequences that could be harmful or non-harmful. And what a mutation is, a change in the amino acid sequence. I mean, in the DNA sequence, which may change the amino acids, which will change the amino acid sequence, which may change the structure, and so forth. So mutations and so forth. Okay, so let's move on. Nucleic sequence, the gene, DNA of an organ is changed as a result of mutations. True. The protein of gene will have an incorrect amino acid. Yes and no. All right. Okay. So, if the DNA sequence changes, okay, yes, you will get. You'll have a new nucleotide sequence, which is slightly different than the previous one, but it may not actually change the amino acid sequence. So if you go back and look at the genetic code, like for proline, the codon for codeine glycine was I think GGG. There was another codon with GGC. So if for some reason there was a mutation that changed that G to a C, you still would get glycine. But sometimes, yes, if the nucleic acid sequence changes, it can change the amino acid sequence. And if it changes the amino acid enough, okay, again, it's not just a blanket statement that you change one amino acid to another, it's going to just blow up the protein. For instance, if you had a mutation that changed a polar amino acid to another polar amino acid, the protein wouldn't care. But if there was a mutation that changed the amino acid to a non-polar amino acid, then it would be a big deal. And that specifically happens like with sickle cell anemia. Just one change, one amino acid sequence changes a polar amino acid to another polar amino acid to a non-polar, and it basically causes the protein to behave in a totally different way than it should. All right, there's different kinds of mutations besides this. So one is called substitution, and that means you have an A replace a T, or a C replace a G, and so forth. There's a picture of it. Okay. And then you can have frame shifts, and these don't happen a lot. And you don't have to worry about these. Just flip through them. There'll probably be multiple choice problems about them. Again, mutations don't have to kill proteins. All right. And here's that. Okay. Genetic diseases. Well, genetic diseases are indeed results of DNA mutations in DNA. So these are mutations that really do some bad things. Okay, so it doesn't just have to be on one piece of DNA. Genetic diseases, there's going to be all kinds of DNAs affected. As we're learning more and more about genetic diseases. So a genetic disease is something that can be passed down from parent to child, because it happens through your zygotes. Okay. And what it is that it changes. If the DNA sequence changes, it changes the amino acid sequence, which changes the protein structure, And if structure doesn't work, the protein function doesn't work. And so you'll see thousands of these. Okay. And here's some lists. Okay, so for instance, PKU, which is phenylketonuria, the ability to make enzyme phenylalanine hydroxylase. No, you don't have to know this, okay? But the kind of curious thing about this is that, and so one way you know you have this disease is that when you go to the bathroom and urinate, your pee is black, okay? And you would figure out real soon if you had this, and if your parents. So I don't know if this is recessive or a dominant gene, but if you had it, you would know that you had it because your parents had it. Okay, and another way you could tell is because you have black urine. All right. Now, so what you want to do, you now if you didn't know you had it and you went on living, you'd probably die because in food, phenylalanine is an essential. It's one of the amino acids. So every time you eat food, you're going to eat phenylalanine. And when you eat phenylalanine, it's going to cause all kinds of problems, and it cause mental retardation and brain damage. So these days, you know that you have it. And so what you do is you make sure that you don't eat a lot of food that has phenylalanine. Well, it kind of turns out it's kind of cool, is that if you look at like Diet Coke or anything that has NutraSweet on it, it'll say contains phenylalanine. Okay, why would Diet Coke contain phenylalanine? Well, that's because it's aspartame. Aspartame is part of aspartame is phenylalanine. All right, and so phenylalanine is not bad. All right, it's not bad for you, but it's bad for these people. Okay, because they can't metabolize it. So they let you know. They put it in big bold letters on the containers that says contains phenylalanine. It's letting these people know that they shouldn't drink it. All right, so here's a list of some genetic diseases. Okay, and people know pretty much the prevalence of having them, and if you have them, you know pretty much if you have a child, your child has a really good chance of having these diseases as well. Okay, mutations. How do mutations happen? Well, they just happen naturally. They're happening all the time just because everything is perfect. But they can also happen through forced ways. And that's being exposed to like chemicals and radiation like sunlight, and x-rays. Okay, so this thing that we just heard about the other day, being blasted with light, okay, that's not a good idea. All right, and that's because is that light is more than just one thing. It's not just sunlight, it's not just blue, green, yellow. It's all kinds of things. Okay, it's X-rays, it's microwaves, it's all right. So I'm not sure what was meant by that saying of just blast with light. Okay, it can be very, very dangerous if you blast with light, especially if you blast with ultraviolet or X-rays. Yes, you probably would kill corona, but you probably kill all your other cells. You cause mutations in them, and then the mutations may cause cancer. Right? So there's some chemicals that we know that cause cancer that are called carcinogens, and here's the list. And so what they do is they just make the DNA not do its job, make DNA. Many chemicals in tobacco are carcinogens, and what they do is it just make actually this is kind of cool how tobacco makes DNA do its wrong, how DNA causes DNA not to make the DNA right, is because the chemicals in tobacco actually make the DNA misread uh, itself and replicate it wrong. So, and then the curious thing is that some cancers are caused by viruses. Okay. All right. So here's the list here. Now, there's things called RNA viruses and DNA viruses. Yeah, you don't have to know that. Okay. All right. Finally, what we have is what we're trying to do these days. We're trying to use this our information of DNA making proteins and the genetic diseases and make better plants and better things and trying to correct genetic diseases um, using genetic engineering. And you can read through this. Again, there might be multiple choices on this. And I think that's about it. I'm going to finish up here. There might be a couple more slides. But again, there'll be a worksheet that goes over the primary parts of this. I know this turned out to be a long PowerPoint, but this is the last one, and there'll be a worksheet about it. Okay, and all right. If I don't talk to you again, good luck with everything. Um, it was good seeing you face to face. We all survived Corona, hopefully, and again, best wishes in your choice field. I hope you move forward. I know all of you in this class will will be getting a C or better, and I appreciate you hanging in there with me um, over these last 5 or 6 weeks.